Good morning, everyone. Welcome you all to this auspicious day for a national seminar on advancements in clinical practice through pharmacogenomics and individualized drug therapy. We all are gathered here today to explore new frontiers and exchange ideas that could potentially reshape our future. Let's start this day with high energy and a round of applause with immense respect and warmth that we are extend our heartfelt welcome to your Honorable Chairman, esteemed Vice Chairman, respected correspondent, respected Dean, Chief Guest, dedicated Principal, Vice Principals, Principals of PGP Group of Educational Institutions, valued faculties, dear students and fellow students from various institutions. Your presence today graces this each one of you. Every note sung in prayers is a step closer to inner peace and divine connection. It's now time for Tamil Thai Valtu.
நிலமடந்த கெழிலொழுகும் சீராரும் வதனமென திகழ்பரத கண்டமிதில் தெற்கனவும் அதிசிறந்த திராவிடனர் திருநாடும் தெற்க சிறு பிழை முதலும் தரிக்கரலும் திலகமுமே அத்திலக வாசனை போல் அனைத்துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிழமை சிரம் வியந்து செயல் மறந்து வாழ்த்துதுமே வாழ்த்துதுமே வாழ்த்துதுமே.
Thank you. Everyone can take your seat. In every weeklet, may our prayer rinse with the light and touch the heavens with gratitude and love as we begin any auspicious occasion by lighting of Kuthuvilakku. I now invite all the dignitaries on the dais to light the Kuthuvilakku.
The best principal don't just lead, they empower others to succeed and grow. We feel cherished to welcome our beloved principal sir, Professor Dr. G. Arunachalam, who continuously works to ensure academic excellence and foster a research-friendly environment to deliver the welcome address.
Very good morning, one and all gathered here. Honorable Chief Guest, the Vice Chancellor, Professor Dr. V. Ravichandran, Delhi Pharmaceutical Research Science and Research University is my Guru Asan. I can say so many things. I am here standing because of him, and respect our correspondent sir and Dean sir and my dear co-principal and Principal SS Institute of Pharmacy, Sangagiri, Professor Dr. Jodi Manivanan sir, and my dear faculty and my dearest students. So today we are gathered here to participate in the national level seminar, Advancement in Clinical Practice through Pharmacogenomics and Individualized Drug Therapy. For this, our resource person, Dr. Sivasangaran Ponnusangar, Professor and Head Department of Pharmacy Practice, JSS College of Pharmacy, Uti, on behalf of Pharmaceutical, PGB College of Pharmaceutical Science and Research Institute, we welcome you, sir. And Dr. A.S. Manjula Devi, Madam, Professor, College of Pharmacy, Sri Ramakrishna Institute of Paramedical Sciences, Coimbatore, on behalf of our PGB College of Pharmaceutical Science and Research Institute, we welcome you, Madam. I heartily welcome once again our chief guest, Dr. V. Ravichandran sir, on behalf of PGB College of Pharmaceutical Science and Research Institute. Welcome you, sir. So with this word, I'll conclude my speech, and I welcome once again all our students. Thank you, one and all.
Thank you, sir. Leadership is not about being in charge. It is about taking care of those in your charge. It is a privilege to welcome our esteemed correspondent, sir, Mr. M. Ganapathy, to deliver his presidential address.
So good morning to all of you. It gives me great pleasure to welcome Dr. Manjula Devi, Dr. Ponnusankar, and more important, welcome back, Dr. Ravichandran, welcome back to him. He's a very, very familiar figure. We had the privilege of hosting him in the past also. And Dr. Ravichandran is very special for us, as the principal has said. He is still a guru of your principal, and he has been a friend, philosopher, and guide to Arunachalam throughout his career. And Dr. Ravichandran is a very eminent figure in the pharmaceutical teaching and training side. Before that, he worked 10 years in a premium institute, National Institute in Calcutta. Now he is the Vice Chancellor of Delhi Pharmaceutical Science and Research University, New Delhi. So today, not only Arunachalam personally, but we are all honored to have Dr. Ravichandran amidst us. My dear students, you should all feel happy and privileged that you have chosen pharmacy as your subject. I tell you the reason why. Before independence, the average age of India was 31 years. When I was a student, school student like you, it was 31 years average age. But today, India, the average age is 71 years. You know, average age means what you call for national average age or longevity. India, 71 years. You know, during my lifetime, I have seen this pendulum going from one extreme to other extreme. There are many reasons for it, but the most important reason is that improvement in the health care in India. Health care is a very, very wide sector, you know, medical treatment and all these things. But in the health care, pharmacy occupies a very, very, very important place, and it will continue to occupy this thing. So pharmacy has played a very important role. I remember when I was a student, pharmacist used to be called compounder in Cholwanga. There was no word called pharmacist. There was a compounder. Compounder is the one who sort of makes a potion and all these things. At the same time now, pharmacist is a respected profession because it is indicative of the fast development of pharmacy, the world. Today, medicine has become part of our life. மருந்தே உணவு அப்படிங்கிற ஒரு ஸ்டேட்டில் country has become, not only India, all over the world, மருந்தே உணவுங்கிற அந்த அளவுக்கு போயிட்டு it's become a part of our daily life. And you can see the manifestation of this thing. இன்னைக்கு பார்த்தீங்கன்னா maximum you go in India, Tamil Nadu particularly, you go anywhere there, Tamil Nadu particularly, you find Iyengar bakery and pharmacy. There was one more thing that has come down now and all these things. So Iyengar bakery and pharmacy, what does it mean? It means, the importance of pharmacy in our daily life and the demand for it also. So that way, that's why I'm telling you, feel happy. You have chosen a very dynamic subject, what I would call is a sunrise industry. Pharmacy is a sunrise industry. It's developing. And India has made a tremendous progress. We are on the top 10 in the pharmacy. And during the COVID, we have seen the remarkable progress India made in the vaccines and all these things. So that's why I told you this thing, and today thing. So feel happy. Only thing is, I would like all of you to advise, having chosen this very important or life-saving profession, you should get fully involved. You should put all your heart and soul into studying the medicine. Studying the medicine. When you pass out, you got so many opportunities of this thing. Try to get into the industry. Try to do something new. And one area in which India has got a tremendous future is the medicinal plants, the local medicines and all these things, which can be developed by the wide variety of medicinal plants in India. So try to be. Padikile, this is what I always say, understand the subject. And there is so much facility available to understand the subject, to improve your knowledge also. Here, let me tell you, the cell phone, the mobile phone, whenever I meet the students, I always call it the good, bad and the ugly. It's the good, it's bad, and it is the ugly also. But as far as I am concerned, I would say there are more good things in this thing. Nowadays, people, patients, when they go to the doctor and all these things, they don't blindly accept whatever the doctor gives. They go and check. They check in the Google, what is this medicine? What is this thing for? And all these. So there is so much. That's why I'm telling you, when you are studying pharmacy, when your teacher explains a concept and all these things, if you don't understand, go to the Google, go and ask and all. There's so much information available. Then enjoy the subject also. Since this is a life-saving subject, it is what I call a sunrise subject. Enjoy reading it and try to, most important thing I want all of you to develop is a curiosity. ஆர்வம் வேணும் படிக்கிறதுலேயும் சப்ஜெக்ட்லேயும். If you have the curiosity, you can do something original. So on this occasion, my dear student, the most important thing, be curious. Try to know what is going on in the world, in your field of study and all these things. Try to be, try to have a sense of purpose of trying to do something new and all these things. So today, I am very glad that Arunachalam has organized this seminar. Let me welcome the students from all the other colleges who are participating in it. Today, that Arunachalam has not only made it a symposium or we has made it a pharmacy festival also. It's about 300 students are here. I think there are another 500 students listening and watching this program. Nearly has made it the whole campus is become a pharmacy day today. So I congratulate Arunachalam and all his dedicated faculty in organizing the seminar. And once again, it gives me great pleasure to welcome Dr. Ravichandran amidst us. He is working at a national level. We are all very happy and proud to have him sitting in the Delhi, being a Vice Chancellor and involved on a very, very national level. So my dear students, so once again, let me tell you, feel happy, feel proud, feel privileged that you are studying a very, very important subject. So I welcome all of you. Thank you.
Thank you, sir. A dean embodies the value of the institution, leading with integrity, vision, and a heart for education. Now I am honored to welcome our energetic and passionate Dean, Sir, Dr. K. Periyasamy, to deliver his felicitation address.
Respected correspondent of PGP Educational Institutions, distinguished guests, Professor Dr. V. Ravichandran, Vice Chancellor, Delhi Pharmaceutical Sciences and Research University, New Delhi, Dr. Sivasankaran Ponnusankar, Professor and Head Department of Pharmacy Practice, JSS College of Pharmacy, Ooty, Dr. A.S. Manjula Devi, Professor, College of Pharmacy, Sri Ramakrishna Institute of Paramedical Sciences, Coimbatore, Dr. Jyoti Manivannan, Principal, SS Institute of Pharmacy, Sangagiri, respected Principal, PGP College of Pharmaceutical Sciences and Research Institute, Vice Principal, respected Principals of other PGP educational institutions, learned members of faculty, participants from other educational institutions, and my dear students, good morning to all of you. At the outset, I would like to congratulate Dr. Arunachalam, Principal of PGP College of Pharmaceutical Science and Research, along with his team of faculty members, for having nicely arranged this national level seminar on advancements in clinical practice through pharmacogenomics and individualized drug therapy. It's our vision, dedication, and unwavering pursuit of knowledge that illuminate the path forward for better, safer, and more personalized healthcare. In recent decades, the field of medicine has witnessed a paradigm shift from a one-size-fits-all model to a more precise, personalized, and predictive approach. At the heart of this transformation lies pharmacogenomics, the study of how an individual's genetic makeup influences their response to drugs. We are fortunate to live in this era where genomics, data science, and bioinformatics have converged to unlock new frontiers in clinical care. As we look ahead, I believe that pharmacogenomics is not just a field, it's a movement towards precision, compassion, and excellence in healthcare. Let this gathering today serve as a beacon for future collaborations, discoveries, and transformations. Let us pledge to harness the power of this science not merely for innovation's sake, but to bring hope, healing, and dignity to every patient we serve. The integration of pharmacogenomics and individualized drug therapy has profoundly impacted clinical practice. In conclusion, the advancements in clinical practice through pharmacogenomics and individualized drug therapy represent a major milestone in the evolution of healthcare. We celebrate the dedication and expertise of healthcare professionals who have pioneered these innovations, improving patient care and outcomes. Let us continue to push the boundaries of personalized medicine, harnessing the power of pharmacogenomics to create a brighter future for patients worldwide. So with these few words, I wish this one-day national seminar all success. Thank you very much.
Thank you, sir. Honoring is a reminds that greatness lies not just in achievement, but in how one touch lives. Now, I would like to invite our correspondent, sir, to honor our chief guest, Professor Dr. V. Ravichandran, sir. Now, I would like to invite our Dean Sir to honor our resource person, Dr. Shiva Shankar Ponnushankar Sir. Now, I would like to invite our Principal of Nursing College, ma'am, to honor our resource person, Dr. A.S. Manjula Devi, ma'am. Now, I would like to invite our Principal, sir, to honor Dr. C. Jodhimani Vannan, Principal, SS Institute of Pharmacy, Sangagiri.
The influence of your good teachers can never be erased, and neither can the wisdom of your great guest. With your great respect, I would like to introduce Professor Dr. V. Ravichandran, Sir, Vice Chancellor, Delhi Pharmaceutical Science and Research University, New Delhi, former Founder Director, NIPER Kolkata, and former Director in charge of NIPER Hajipur, having 28 years of teaching, research and administrative experience. His research is focused to utilization of animal models for evaluation on efficacy of natural products and understanding its mechanism of action, development of pharmaceutical products from the natural source and semi-synthetic process development for making affordable medicines. He is passionate about translating the results of his research into products. He has utilized various techniques including pharmacological models, molecular and cellular regenerative medicines, multi-omics, CRIA, SPR, CAS system and imaging technologies to answer his research work problems. He is instrumental for setting 3 Center of Excellence such as continuous manufacturing, biological therapeutics and marine therapeutics. He is actively involved as pharmaceutical consultant for mid-sized pharmaceutical companies. Under his guidance, the faculty developed oral complex dosage forms for the management of IBD within 2 years span of time. He has published 162 publications with 15 patents. He mentored 21 PhD scholars and 3 postdoctoral candidates. He has completed his 5 projects founded by AICTE, ICMR, DST and DPT. He is leading a multi-institutional project center of marine therapeutics as a center coordinator and principal investigator, which is jointly funded by DST and Department of Pharmaceuticals. He is established Center of Nucleic Acid Therapeutic Process Development funded Chemical Therapeutics Program of DST to treat DMT rare disease. He has been involved in development of various animal models of human disease for pharmacological screening of various drug candidates and natural products including our Indian traditional medicine, and peptide mapping too has been utilized to generate evidence-based medicines from natural source and ensure the efficacy of phytomedicine therapy. I feel privileged to invite our chief guest, Dr. V. Ravichandran sir, to share his insights.
அனைவருக்கும் வணக்கம். president of the today meeting, Dr. Ganapati, the correspondent of PGP Group of Educational Institution, the chairman, PG Periyasamy, the vice chairman, Visalakshi Periyasamy, the principal, Professor Arunachalam, the dean, K. Periyasamy, and all other, the head of the institutions of different school, invited speakers, Professor Ponnusankar from JSS College of Pharmacy, and Dr. Manjula Devi from Ramakrishna Institute of Paramedical Sciences, Professor Jyoti Manivannan, our good friend, all our faculty, young friends of pharmacy and different pharmaceutical institute, participants from various organizations. I am really delighted to be part of this today event. Advancement in clinical practice through the pharmacogenomics and precision medicine or that individualized drug therapy. It's a very important concept. The principal, Professor Arunachalam, has identified and for which the speakers are here with the high credential and expertise in this area. Professor Ponnusankar and Professor Manjula Devi is with us. JSS College of Pharmacy, pharmacy practice is one of the very pioneer in the pharmacy practice area. So, the Ponnusankar is invited here to share his expertise in the area of pharmacy practice, how it would be useful for that improving your skill. The same manner, Professor Manjula Devi, in the Ramakrishna Institute, another important pioneering institute in pharmaceutical sciences, which add value for that the pharmacy practice area. This gives that inspiration for that young mind, what to learn, what to do on this. You may think about that, what are the pharmacogenomics and personalized medicine or individualized drug therapy? Being you all here in the pharmaceutical science area, you should understand what is our preparedness. You are joined in the program course, you are going to get the degree program, you are going to get qualified. Meanwhile, what are all the serious component? You have to think about that. More than 1,000+ concepts and problems are there in the society. Identifying the problem is one of the serious concern. The problems are identified, then you should read regularly for this purpose, understanding the problem. Understanding the problem. What understand the problem, how you understand the problem? Unless otherwise, if you collect some analytics data on that in the society, what kind of the problem in the health area it is there. Whether one pharmacist or one doctor can address all the problem, it is a very challenging component. That is the reason earlier it was a practice, the conventional practice more than 100 years before. A doctor, generalized doctor, he will treat all the problems. Now the case is, currently it is not in the level. All specialized doctor for each and every problem, even for that wound healing. skin doctor, eye doctor, nephrologist. In this case, there are several segments of that the doctors are here. The pharmacy practice in the same manner, it was not earlier with that 50 years before or 100 years before. As rightly conveyed by that our president of the program, Dr. Ganapati. See, the problem is that you have to think about how the life longevity. 50 years before, what was the status in our country, and 100 years before, what was the status in our country. Now currently, what is the status? Current status is that more than 70 years, minimum 70 years. This is what Dr. Correspondent was pointing out, the longevity of the life. The purpose of that indicating how we are going to prepare ourselves. This is the main component. The seriousness, we know about the protein. The protein has that individualized function. While we read about the protein, enzyme it comes, some of the metabolic cycle it comes, we will keep all in the choice of that. We will not touch this thing. This is always we will keep it in mind that no, this is the enzyme and metabolism. These are all the complex area. We need not worry about this thing. We will have that some choice on that. But somebody who focused manner understand that what is a protein, what is its function, and protein expression it is more, protein expression is less. What happens? This is what disease condition. Physiology, if you want to become an expert. Yesterday there was a program for the Vikshit Bharat, roadmap for that, preparing manpower for the global demand. Preparing manpower for the global demand and global supply. You are all going to be a leader after 20 years. While country independence, it goes to that 100 years. 100 years of that independence. The India is expecting to be the leader of the global market with the knowledge power. The knowledge power, how that the knowledge power it comes? Not by the choice. You have to face that the question. You have to understand that the question. You have to address the problem. By this way only it could be possible. That is the main reason your principal has identified pharmacogenomics and personalized medicine, individualized drug therapy. In the conventional practice, normally the drugs, what it is approved drugs are available in the market. These drugs are not suitable for that all 100% of the population or the patients in the chronic cases. The drugs which is used for that certain chronic problem or any of the ailment, 70% of the prescription, the drug is useful with side effect, with the adverse reaction, and some of the patient it is not effective, and some of the patient it will be very toxic, in some of the patient non-toxic, and there is no effect at all. So in this the complexity current practice and the therapy management is happening. How to address the current problem? The current problem can be addressed only by that understanding that pharmacogenomics. You should be very clear about that. What is disease? Gene defect protein imbalance, gene defect by heredity, the gene disorder, this is a rare disease. Environmental disorder, epigenetic modification, the genes are modified, epigenetic modification, methylation, acetylation, chromatin modification, histone modification, chromatin modification. By this way, the disease is that developed, the pathogenicity it comes. Once methylation to demethylation, acetylation to deacetylation, it is a regular physiological function. If the demethylation, deacetylation doesn't happen, disease starts. You have to be very clear about that. When the problem starts, the repairing system doesn't work, then disease starts. You should understand, I believe that you all might have gone through that. What is pharmacogenomics? Why they have given that the title? You should question yourself. You are not done. You go and see that. What is gene? What is gene therapy, cell therapy, epigenetic modification, methylation, acetylation, demethylation, deacetylation, methyl transferase, acetyl transferase? What happened that the histone modification and the chromatin modification, it happens, drug works or drug doesn't work. Where is the problem here it comes? The disease starts with that the inflammation. It goes to inflammatory disorder, metabolic disorder. Metabolic disorder, it goes to that the fibrosis. Fibrosis, it goes to that necrosis. Necrosis progress into the cancer level. The cancer doesn't come somewhere else. That the problem starts with that smaller level, with the simple pain, and progress into different stages of inflammatory disorders, metabolic disorder, liver function, mitochondrial dysfunction, autophage dysfunction. This all happens to reach that the problem of that pathogenicity. The pathogenicity with that cross-linking damage with that fibrosis, necrosis, it gives that ground for that the cancer. You have to be very clear. And what I want to say, every one of you, you cannot be a master in that 1,000+ concept. Whatever the designed program of that the degree, it is well framed. You have to read, understand, and perform, appear for the question. You have to pass that the degree. This is one of the component. You have to develop your skill. I am qualifying the degree. That is the examination for qualifying the degree. That is not the skill. This is one of the basic component for establishing yourself, performing, and proving your capability of that the understanding and presenting, reproducing, and performing. With that the score you are getting 35%, 50%, 60%, 75%. You are proving, you are passing the degree. Whether this is sufficient for ensure your capability, this is not sufficient to prove your capability. Capability still it need that skilling, upskilling, more specialized skilling. Here it comes into that the part of that the protein. Gene is responsible for that expression of the protein. Each protein has that specialized function. These that the function it is, protein function is modified, that become that the disease. You got the point? It's a very serious concern. Why I am trying to repeat that the component, you have to make it clear what you are going to do for that the future. Vikshit Bharat, the country, policy, the government, the Prime Minister, Honorable Narendra Modi, he is looking into that. Our country, the knowledge power should supply to do that the service, highly specialized service to that the other country of 220 countries it is available. What the way and mean that currently the vaccines, It is supported by India. More than that, the 70 countries it is supported by India. This is what it is happening from that the India to other country. The same manner, the knowledge power is very serious concern. The knowledge power, by the way of the knowledge, we can do service to the other country. This is the Vikshi Bharat. The manpower development, knowledge development with the multidisciplinary aspect is a serious concern. The roadmap means today if you prepare that yourself, it looks you are very simple and small boy. Sir, I am a small person. My parents, they are putting some money and they admitted here. That is a fact. No one can deny it. You will become that wonderful leader after 15 years. I was a simple person, very simple person, 30 years before. Sitting there like you, so what we are going to do? While my teacher was asking me, I said, you recommend me for a company. Then he was suggesting, no, the future is very best. What future is very best? If I get 1,000 rupees, that is a very big thing. That I am saying, during 1990, I was getting 750 rupees. The reason you should understand that. 750 rupees, for the 750 rupees, what you will do today? You will have one lunch. That is the entire 1 month salary. I was working for a company, IDPL, Indian Pharmaceutical, Indian Drugs and Pharmaceutical Limited, IDPL company. So later on, I joined the postgraduate, and these are all the simple components, simple way and mean that developing ourselves. The reason why I am narrating that, my service during 30 years before and that the education in the postgraduate now currently, these all periodically how you are performing. Every day you have to read and read. What is that the reading? Reading, what it does? Reading gives that confidence. Reading gives that courage. Reading gives that performance. Reading gives that perfection. Reading gives that dynamism. You will be very confident in front of anybody if you know that answer. If you understand that what is gene, what is genomics, then you will be very courageous. While seeing of this paper, once that the circulation it comes, we understand that something pharmacogenomics, what it is, I don't know. You also don't know, I also don't know that, but we should learn what is this pharmacogenomics. Then only it gives that confidence. Yes, this is what it is, the genomics. This is what a drug does into that the gene, and expression of that the protein it is modified. I believe you all know that remdesivir. It is the 4 years before. The COVID was that the major challenge. In everywhere that the people were aspiring to get that remdesivir injection. What is the reason on that remdesivir injection? Any one of you know that what is remdesivir and what is the objective of this remdesivir injection? This is a very serious concern. We should not that unaware about that. This will not give you that any confidence. This will not give that anything. The courage for that working in a company. If you don't understand, then how the things will happen? The remdesivir, very simple manner I tell you that. Gene is there. The gene, there is a transcription. There is a transcription, transfection, translation. There is a protein expression. It comes from the gene only. The DNA to RNA, RNA to protein. This is, I believe, in the school itself you might have studied. Otherwise also you go and see that. Because the protein is such a serious component. Protein is, it is not that the simple thing. If you are always thinking of that, no, what is this protein? The COVID, AC2, which modifies certain proteins, that become the disease. Expression of this, that the COVID by RDRP, the proliferation of that the virus is a very serious concern. If RDRP is controlled by remdesivir in the transcription, protein is that not expressed, then that the load of the virus may not be increased. That is the purpose. Remdesivir is the one of the serious medicine. The people were rushing for the injection to control the viral load. That is what the protein, it is called as an RDRP. In all the virus, this RDRP is one of the segment. If the RDRP, if it is controlled, then viral load would be normal. Immune system control the problem of that, the virus. Then there won't be any problem. That is the reason people, everyone want to save that their parents, their brother and sisters, all the family members who are affected with the COVID. This was a big challenge. We should understand that we should prepare ourselves. Yes, if such a situation in the future it comes, how to do, how to manage it. We have to very seriously understand that the single problem of 1,000 plus concept and hypothesis, the questions are there. You have to select one question and equip yourself in the knowledge, wisdom. This the wisdom gives that platform. This the wisdom gives that the courage. Yes, I can talk for that 1 hour about this protein. I was telling you that the simple problem only it starts, end with the cancer. We should see that how the disease is progressed, how that the simple problem of the pain, metabolic disorder, it goes into that the several segment of complexity and end with the cancer. Then the cancer, if it is a preliminary stage, it could be controlled. And the cancer is, it is in the matured stage, level 3, metastasis, it cannot be controlled. Still, there is the possibilities of that antibody drug conjugate. These are the things it is associated with pharmacogenomics, that is a personalized, individualized drug therapy. It is what the tradition. Traditionally, one drug it is available, if it is generally it is given, it goes into that, for somebody to work, somebody it doesn't work, it makes resistance or it causes that the adverse reaction, toxicity. That is the main reason. Personalized medicine, you have to study that the human genome. With that the human genome, pharmacogenomics, how that drugs works with that the gene, whether it works or it which is resistant, sensitive or resistant. This is a very serious concern. If you don't understand as a pharmacist in the pharmacy practice, if you are specialized with one problem, the problem can be infection, problem can be metabolic disorder, problem can be fibrosis, liver fibrosis, fatty liver, or that the renal failure. The problem can be that neurological disorder. It can be with that Alzheimer's disease or Parkinsonism. Whatever it is that the issue, I am again telling you that there are 1,000 plus problems. One individual cannot treat that all the problem. You cannot be expert in all the problem. That is the purpose of this, that pharmacogenomics and individualized drug. Even I am trying to tell you that you target that 6 months. You understand one problem. with the make that the group of that 5 and 6 people, and segment wise you make that this is a 10 assay it is available. In the morning I was sharing with Professor Ponnusankar, if you understand that one assay, you will pass the semester. If you understand that the 3 assay, you will pass first year. If you understand individually, you can conduct one experiment after that the 4 years period. You will conduct that one experiment in which that the 3 segment is there. This is a profiling, this is a characterization, this is a imaging, this is what estimation, qualitative analysis, quantitative analysis. This is a current data on this. This together I am submitting one paper, and these such kind of the things, if you do it in the final year, 3rd year itself, if you get the 2 paper, you will get that wonderful job in the final year itself. That is a placement, training and placement. Preparedness for that placement and training, for which this is the pharmacogenomics and individualized drug therapy will be useful. You should not take it very easy. You have to be very sincere and serious. In a day, if you read minimum 5 hours, you will become a wonderful personality with wisdom, with high skill and developed skill, with credential. These all will give you that several design, because these are reading and reading and reading, collecting the analytics from that the different paper of that research paper like science, the nature. There are several components. These are more than 100 plus years. I can say that the 400 years before that, 18th century, C.V. Raman from here in the country India and the south Tamil Nadu, he was there in Bengal, 18th century. What he did that he got the Nobel Prize, and C.V. Raman Science Day, now that still entire world is celebrating. He was a normal person here. He was an accountant there in Bengal, Calcutta. He was a accountant. He was not a scientific person. He was working in the accountant area. You go and read, you can understand that. Now that Raman spectra, the Raman spectra, you can understand that why the Raman spectra you are talking. Its relevance is there with that pharmacogenomics, what it is there. Raman spectra, with the Raman spectra, transcriptomic can be studied. That's what in the 18th century, what he developed in a small lab in his house, individually. Then later on that, he worked in the Indian Association of Cultivation Science in Calcutta. You see that he was working in the accounting area, and the Raman spectra, he was developing that the Raman spectra concept, principle, instrument. Now this Raman spectra equipment cost, you know that minimum of 2 crore and 50 lakhs rupees, these are the equipment, it is everywhere in the entire world it is there. So the concept would be that even while he was learning and reading, he was a simple person. So you have to believe on you. If you don't believe on you, who will believe you? This is a very serious component. Another important aspect, it's connected with that the pharmacogenomics. The pharmacogenomics, you have to see what are the different drugs are available in this area of that personalized medicine. You may think that where is this kind of the practice? This practice is here in India itself. The pharmacogenomics, personalized medicine, this practice is there in India, but it is 0.01% in the tertiary care area, very highly wealthy people who can spend more money with that group of doctors and the clinical pharmacists, they are working for that and giving the treatment. This is not happen that currently major level higher percentage, but the future is personalized medicine. The people, they don't want to have the trial. Okay, you take medicine A, medicine B, medicine A and B is not working for a 1 month period, medicine C will be given that after 1 month. These are the days are outdated. Now currently you have to prepare yourself. This is the drug, it is sensitive, it works. For which there is a model of that organoid. Even I was talking with Dr. Punisankar for the purpose of that, how organoid can be developed to study that the drug sensitivity and drug resistance. A problem of that, the fatty liver, a problem of that hepatic cancer, a problem of renal cancer, how that antibody cost of that more than 50,000 rupees, even it goes to 1.5 lakhs also. If it doesn't work, if the 3 injection it is given, 4.5 lakhs to 5 lakhs of the money, whether you will get back the 5 lakhs of the money, if you have the skill for that identifying PBMC, IPC for making of this thing, either with that liquid biopsy or that solid biopsy, taking and developing within a month period, you can develop this primary culture and organoid. If you have the skill, if you have the patience, if you have the wisdom, it could be possible to develop. And if it is tested, the doctor will respect you that, yes, this is what I need. I don't want to give that itna injection, 5-6 injection or 50 injection, more than 15-20 lakhs spending money. Later on that the drug doesn't work for this patient, he died. This is not going to happen in the future. You have to prepare your mind very seriously. I don't expect that all of you can understand. But even 10 member, 10 scholar, if you understand that the importance of the concept, it gives you that wonderful, the segment of development, the credential. So the proteins, anything epigenetic component, what you are everyday eating, it's the environment, what you are eating the fruit, what you are eating the food, what is the environment in the water and then the atmospheric air, which modify the gene. That is the epigenetic modification. That is basic concept is methylation and acetylation, which shown in that the protein expression. Anything food you are consuming or medicine you are consuming or a natural product or a drug or a Ayurvedic drug you are consuming or you are subjected, which makes that change in the protein expression. The same thing what it is that the virus or fungus, any infection it happen, or that any pesticide or that any chemical it is subjected, poison it is that is subjected, it affect that the gene expression of the protein would be visible. That is what the protein expression, upregulation and downregulation, it could be possible to understand by proteomics and peptide mapping. This is assay system. You should understand that how to conduct that proteomics, how to conduct that gene mapping and peptide mapping. These are that normal physiology, this is the pathology. In this condition, after that subjecting this drug, this is the evidence of that the protein expression it has come. That is the area now that is emerging. It's a proteomics. Pharmacogenomics, that's what the principal was saying and talking about the pharmacogenomics. Omics is that the futuristic aspect, it is very serious concern. You have to understand about all the omics, either it may be that transcriptomic or that pharmacogenomics or proteomics, all the omics are that now evidence based. This analytics, What you have to do, you go and read about that omics. What is this omics? What it sounds? What is the objective? Why this all that is called as an omics? Why we should learn this? What is the use of this omics? How we can learn this wisdom would be useful for us talking the science, pharmaceutical science. How that pharmaceutical intervention in that epigenetic management which solves certain problems. So these are very serious concerns. You have to think about that. Being a teacher, I can continuously talk on this, the topic, more than hour plus, but there are specialized, the speakers are available here. They will talk about that, the topic, the concerned topic. If any of you think that, no, what I couldn't understand what it is, you can write to me also, you talk with your principal also. I am very happy that to help you on this, but these are very serious component. One word, what it is that pharmacogenomics, it is written, and individualized drug therapy. What is the problem is there? Why individualized drug therapy it is need? If you understand, then it develop inspiration with your mind. This is a very serious requirement. Inspiration in the mind is very important for learning certain things. You have to question yourself and what it is written. Then you will get that more idea. With this the few word, I believe that the talk for the today, it is going to give you that good thought, good message. You can read for that next 1 year period continuously. If possible, next year also I will come. Because this is at the institute, regularly I am coming. This is not the first time I am coming. Almost 15 years before, I was here that regular, every year I used to come. Because of that, I was there in that Calcutta, Bengal. I could not able to come that. Then later on, few years back, I came here. Now I am here that. I am very happy that to meet you all through that your organization, institute, the PGP. They are doing the wonderful job. That is the reason they called that Professor Punnusankar and Professor Manjula Devi, what they say that very carefully listen. Discipline is very serious concern for learning. You have to keep your mind with attention. There should not be any diversion. We have to listen what this gentleman is saying. Then only you can understand. If you get diverted in different direction, we can simply laugh and go. Your parent hard-earned money, a very serious concern. This is not very simple thing. That today, what you are spending, even it is 1,000 rupees, this 1,000 rupees is very serious concern. Correct? You should think that today we entered here for this purpose. We should not waste that this 1,000 rupees hard-earned money of our parent. If this the thought comes, today you will learn more. You learn that equal to 10,000 and 1 lakh rupee, it will give inspiration for several million. So this is a very serious message for you all. That's the reason, in this the case only, I am sitting there in Delhi. It is not that with any other component. My father was a very simple farmer. I am sitting in Delhi as a Vice Chancellor. You should understand that only the wisdom and confidence on the wisdom, self-confidence on that learning and competing, competence on that, the talking with anybody that he may be a doctor or he may be a scientist. It's a competence, it's a very serious concern. If you don't know, you should say that I don't know. But I will learn and come back. I know that about pharmaceutical application. This is a molecular science. I have to understand further because this is a learning. If you learn such a way, you will become that wonderful person. I stop here. I thank the organizing committee and the management for that, the best way of practicing all the young people for preparing that the Vikshit Bharat, you are going to be the leader. You should think that I am going to be the leader after 20 years. I will go to that, I will go to the Pittsburgh or I will go to that Max Planck, I will go to that Fidelbia. These all I am saying that globally recognized university, then you can understand that it gives that confidence. That is a success of that the learning. You don't think that it is a, in the country, 78.6% of the scholars studying in the self-financing college. This is the reality of the country. 78.6% of the students are studying in the self-financing college. Country is the poor country. If it does not happen, then you cannot qualify yourself. Only the confidence. Today you can sit here, tomorrow you can sit in the IIT or a national institute in the pharmaceutical education and research, or even in the DIPSR. So you have to have the confidence. We lost yesterday, we gained today, so tomorrow we should be there in that competing with all other students, not only with laughing you can go there. Enrich your knowledge and the wisdom and the skill, it will give confidence, you will be that the winner on this at the competition. All the best for you, and thanks for the organizing committee, the best way that the guiding the teachers and the students for that the winning path. Thank you.
Thank you, sir, for your insights on pharmacogenomics and individualized medicine. I would like to welcome our dignitaries from the dais to the seat.
Your presence is not just an honor, it's an inspiration to all of us gathered.
I would like to welcome our dignitaries from the dais to the seat.
outset, I take this opportunity to thank the organizers for inviting me to share some of my research experiences with our research team at the Jesus College of Pharmacy, Ooty. And I also thank my senior, Professor V. Ravichandran sir, for recommending me to deliver my research experiences with the young minds. And thanks goes to Principal Dr. Arunachalam. Probably this is the first time I'm meeting him here on the campus. Of course, I am nothing to do with pharmacy practice. I'm an M.Pharm Pharmaceutics guy. I did my M.Pharm Pharmaceutics. And out of my passion, I initiated this department way back in 1996 at Jesus College Pharmacy, Ooty, and Dr. Ravichandran witnessed that. And over 30 years of experience and commitment and collaboration with my counterparts at Australia, we consistently performing in training our PharmD graduates as well as M.Pharm pharmacy practice students. In fact, when you see the pharmacy practice research, it is totally focused on patient care. It starts from conducting a survey. It starts from doing the drug utilization evaluation. It starts with drug information services. Sometime different type of research which moves from patient-based or a group of patients. But rather during the practice, we observed that many times the individual patient's therapy is failure because of various reasons. You all know that in Ooty, being agricultural land, a lot of agrochemicals were used for the cultivation of carrots, potatoes. Of course, for the production, mass production, definitely we need agrochemical. But often, when these patients come to hospital with a lot of problem, we could not manage them. So my area of interest is genomics, and initially we have started the study of us understanding the inter-individual variability of diabetic patient management. And being a public hospital where we practice, it is a very limited resource setting hospital it is. So we thought that why don't we initiate the services in our department and the colleges, the college premises. So we initiated this research area. And now I'm slowly moving from the genomic area and especially psychiatric, where we get a lot of problem. And maybe in another 15-20 years time, we'll be having more number of psychiatric patients in the country. The reason which I foresee is like we'll be having more elderly population, and they'll be alone. And they get a lot of psychiatric issues. And this presentation introduces the paradigm shift in psychiatric treatment towards personalized medicine, particularly focusing on precision psychiatry. It also explores how polygenic insights like polygenic risk score and cutting-edge diagnostic tools like CRISPR, Cas13a, which are going to revolutionize the antidepressant therapy. The approach, what we are going to discuss today, is like of how the inter-individual variability in drug response, offering a targeted framework based on genetic, epigenetic, and pharmacokinetic data. Before we go with that, let me introduce what is precision psychiatry. Today, during the presentation, Dr. Ravichandran sir told about individualized therapy. Of course, it is part of that. Precision medicine, which many of us would have heard about it. So in the part of personalized medicine, we would like to talk more about the precision psychiatry. It is a modern approach to mental health, especially mental health care that uses individualized biological, genetic, and environmental data to tailor psychiatric diagnosis, treatment, and prevention strategies to each person. Instead of relying solely on symptom-based classification, as in traditional psychiatric management, precision psychiatry incorporates omics-based science, which includes genomics, proteomics, metabolomics. It's all included in that, and digital phenotyping and clinical biomarkers to guide decision-making. What it matters in precision psychiatry? In fact, the core principle acknowledges that one size fits all. One size doesn't fits to all, and it is the way how we take the understanding of the treatment don't work for all the patients. Of course, it seeks to understand the underlying biological variability in psychiatric illnesses. It focuses on proactive, predictive, and preventive care, not just reactive treatment. How pharmacogenomics supports precision psychiatry. And precision psychiatry powered by pharmacogenomics is transforming mental health from a generalized approach that is tailored, targeted, and biologically informed. It also offers an opportunity to improve the outcomes in difficult to treat conditions like treatment-resistant depression. That's where we are trying to master. So treatment-resistant depression is the one problem which we have come across majority of our Indian population, because the treatment is given only based on trial and error method. Once the treatment is initiated, as I said, one drug will be given to the patient, and sometimes patient will get remission, relapse, and patient after 8 months' time he will come back to hospital with no benefit. Then one more drug is initiated, either the dose of the same drug is increased or another drug is given to the patient, and after 8 months' time or 10 months' time, again the same problem. So without understanding the genetic variations, which influences the response of antidepressants, antipsychotics, and mood stabilizers, we cannot initiate the therapy. So precisely, we would like to introduce the drug to the individual patient and treat individually. And why we need to make the effect of precision psychiatry? Because it reduces the adverse reactions and non-adherence. And very genetically, if we think about this, we have few examples, which includes the individual patient who has a poor metabolism. He may accumulate serum serotonin reuptake inhibitors, leading to side effects. And we need to initiate gene tests like gene site and CNS dose, which is guided for the medication choice. What are the applications of pharmacogenomics in psychiatry? High variability in treatment response, 30 to 50% of the patients do not respond to the first-line antidepressants. And adverse reactions are very common, which are leading to poor adherence. Traditional psychiatric care is based on trial and error prescribing, but many psychiatric conditions like depression, anxiety, schizophrenia are complex and heterogeneous in their biological basis. Hence, personalized antidepressant therapy is initiated. And for an example, we need to take an example of poor metabolysis with the CYP-C2C19. And when they are poor metabolysis, they avoid drugs like citalopram and sertraline. Similarly, patients with SLC6A4, which is a short genotype, where they have less responsive to serum serotonin reuptake inhibitors, we have to consider non-SSRIs. The next point which we should consider is prevention and early intervention. Individuals with high polygenic risk scores for major depressive disorder or treatment-resistant disorder, diseases can be monitored very closely, and we can give early behavioral therapy or non-SSRI treatments. And of course, many times we will end up with treatment failure. We need to identify whether the patients are poor metabolizers or fast metabolizers. What are the key genes involved in psychiatric pharmacogenomics as of Indian population? Where for the metabolism, CYP2D6 and CYP2C19, and we all know that for the metabolism of any product, any drug molecule, we need cytochrome P450 enzyme, CYP3A4 and 2D6. And for the antipsychotics, as well as all psychiatric medications, the individual required CYP2C19 isoenzyme. Of course, if any problem in genetically, and it will affect the antidepressants concentration in the body, whereby patients won't get any therapeutic benefit. Similarly, there are many proteins, which Sir was discussing today morning, protein. So SLC6A4, and this is the target protein for us, for the Indian population, sir. Our study is entirely based on, for the especially South Indian population, we target on SLC6A4 protein. And there are many other proteins expressed in the body, which are also responsible for various therapeutic benefit in the Indian population. What are the drug concepts of drug response modification, and what are the factors which influence the drug response in Indian population, which includes pharmacokinetics, dynamics, transporters, and HLA-related toxicity. And each and every concept should be taken into consideration. And we need to train ourselves beyond the pharmacy practice. We need to train, and there's a big future in working in this area, especially in the research area. The kinetics, of course, my colleague Dr. K.P. Arun is involved in developing the population pharmacokinetic concept at our institute, where he trained many students from PharmD background, pharmaceutics background, and mathematical modeling of establishing the dose of a particular drug. Usually, the trial and error method is given to treat the patient condition. But before we initiate that, we can make the mathematical modeling, and then we can, yeah. Yep. Thank you, sir. We can create a mathematical modeling of dosing of a particular drug, which is maybe very expensive, or this drug, to avoid therapeutic failure that can be used. And the pharmacodynamics we learn more in pharmacology, as well as we should learn more about the transporters and accelerated toxicity also. The first domain which I would like to introduce out to is to understand the therapeutic failure, or to prevent the therapeutic failure, we need to initiate the polygenic risk score. Polygenic risk score is a quantitative score that estimates an individual's genetic predisposition to a complex trait or disease by aggregating the effects of many genetic variants, usually single nucleotide polymorphisms across the genome. Unlike the monogenic diseases caused by mutations in single gene, monogenic disease which include sickle cell anemia, sickle disease is very common with tribal population where they are living in Uti. But there is a polygenic disease like diabetes, depressive disorders, schizophrenia, generalized anxiety disorders. It's very, very common. If we do the survey of generalized anxiety disorders among this population, I'm very sure that 50 to 60% of all of us are generalized anxiety disorder will be having it. So we should try to understand why it is occurring, the epigenetic reasons for that, and then we try to initiate the treatment for that. So the polygenic meaning that result from the combined influence of hundreds of thousands of small epigenetic variants. And of course, the key features of polygenic risk score is, includes based on genome-wide association studies, from there we can take the data. Of course, the Indian data is also available. And this data is very useful for complex polygenic disorders like schizophrenia, depression, diabetes, etc. And based on this data, we can do the polygenic risk score assessment by this formula. And the scientists first look at the large genetic studies that identify many genetic variants associated with the diseases. And each of these variants has a weight of effect size that shows how much it contributes to the risk. And the PRS formula is calculated. Of course, for Indian population also, we have genome-wide large genetic data. For Indian population also. From there we can take the data and it can be applied to find out the sum of PRS. And this is the way it is calculated, the steps involved in the PRS calculation. And we have to identify the single nucleotide polymorphism associated with the trait. And the second step is to identify the SNP selection, single nucleotide polymorphism. And third step is weight assignment. And fourth step is to do the PRS calculation. And last step is to do the calculation. By doing these 5 steps, we can identify the PRS score for the individual person, and we can identify that which drug is going to be useful for this individual patient genetically, and we can avoid the treatment failure. But what is the burden of antidepressant non-response in Indian populations? Around 50% of Indian patients do not respond to first-line antidepressants, leading to treatment-resistant depression, defined by the inadequate response to 2 antidepressants. And because this condition increases suicide risk and healthcare burden, assume that in our home, if you have an elderly person who is getting treatment with psychiatry, and by giving some medicines, if the treatment is not continuous or the patients are getting benefit, definitely the suicide thought will increase and will be losing their family member at the home. So understanding its biological basis is key to early intervention, and definitely the antidepressant therapy should be initiated based on that. And the treatment-resistant depression is a big problem in India, especially with the elderly population. It is a major depressive disorder that fails to respond to at least 2 adequate trials of antidepressant. 2 trials, that means first drug is given, let us say sertraline is given, first 8 months, patient will get some benefit, and after 5th month time, patient won't get any benefit at all. And again, after 15th or 16th month, one more drug will be initiated, like this trial will going on. So within 24 months time, patient will decide that these drugs are going to help, so his suicidal thoughts will increase. So 2 different pharmacological classes, each of sufficient dose and duration, usually more than 6 weeks is given. Whenever a treatment to depression or anxiety is provided, 1/3 of the people show complete remission, another 1/3 show response but no remission, but the remaining population show a complete resistance. So this is the 2/3 of the people with no remission or susceptible to be TRD individuals. What are the contributing factors for the TRD, treatment-resistant depression? First one is biological, because of gene variants. We have seen high variation in the gene pattern across India. And the next one is which yesterday myself and Professor Ravichandran were discussing about the neurotransmitter imbalance. That is very, very common with the elderly population. Because of mood elevation, a lot of problem is observed. And many times it's observed because they are alone at home. Nobody is there to take care of them. The second reason is psychosocial, and of course it's associated with the biological, stress and trauma. And third one is clinical conditions because of comorbid conditions, misdiagnosis, and non-adherence of the patient. And these multifactorial causes necessitate a comprehensive diagnostic model. And if you see the pathophysiology of the treatment-resistant depression, it happens when a person's depression doesn't improve after trying standard antidepressants. And this is not due to one of the single issue, but rather several complex and interconnected problems in the brain and bodies involved, which includes mononergic system, neuroplasticity, and HP axis regulation, neuroinflammation, because of various reasons. But if you see the clinical presentation, the impact of the TRD, which includes persistent low mood, then anhedonia, fatigue, and cognitive impairment, often more chronic, refractory, and disabling, and high healthcare utilization because they repeatedly come back to hospital again and again to get the treatment, and the cost of the treatment has become more higher. And not many populations are affordable to do that. And treatment dropout, because of that reason, they won't take the treatment continuously, and increased suicidal thoughts. That leads to a big problem and responsive depression. And what are the conventional treatment strategies which we have? So we need to switch the antidepressants frequently, every 6 weeks once, which patient may not benefit it. And at one point of time, the cost of treatment is going to be expensive. And augmentation therapy with lithium, obviously it's a neurotherapeutic index drugs. We need to fix up the dose based on the patient response rate, and it's a very expensive procedure. And combination therapy, which includes a serum serotonin reuptake inhibitors and bupropion, mirtazapine, or tricyclic antidepressants, and non-pharmacological options like electroconvulsive therapy or repetitive transcranial magnetic stimulation or psychotherapy. Many patients will go with psychotherapy advice, but which includes switching of drugs, combining medications, or augmenting therapy with lithium or antipsychotic therapies, but non-pharmacological therapy, sometimes some patients will get benefit out of it. But the precision of process which we are focusing more is on developing the treatment, selecting the treatment based on the pharmacogenomic data which is available in the Indian population. And obviously, we need to identify whether the patient is poor metabolizer or non-responders based on the gene pattern. And that leads us to, that give us an opportunity to select the drug and dose adjustment also. At the same time, we should also try to understand the genetic polymorphisms and drug response. There are 4 different domains which we need to focus more on selection of genetic polymorphisms. One is poor metabolizers, where the drug will be cleared very slowly, drug clearance will be very slow, so whereby drug levels will be increased, whereby patient experience side effects. The second domain is ultra-rapid metabolizers. Through genetically we identify them, and they'll do the fast clearance, drug levels will be lower, therapeutic failure is observed. Other side, patients will have a problem of absorption of a drug through ABCB1 glycoprotein transporter, where we will find increased activity of the protein, whereby low drug concentration in the brain, where patient will experience poor efficacy of the drug. And the next domain is reduced activity of the glycoprotein transporter, where higher brain drug levels are observed, improved or toxic response. And the one what we have learned so far is the tip of the iceberg of pharmacodynamic genes, which we are focusing with the southern population only on SLC6A protein. And the next major protein which is involved in this HTR2A protein. So these 2 proteins we are trying to encode it in Indian population, and we are trying to titrate the dose of the drug and sex of the drug also. And very importantly, in India, we found a lot of epigenetic changes which on drug responses. There are a lot of studies available in Indian population, especially southern populations, which is focused on DNA methylation, histone modification, and microRNAs, which Professor Rajendran was discussing about it today during his talk. So all these epigenetic changes, which are directly related to the responses, we need to take care of that one also in fixing the dose as well as of the drug also. While talking about the clinical integration of genetic and epigenetic data, the combining genetic and epigenetic data provides a fuller picture. Environmental influences, stress and drug exposure can alter gene expression via epigenetics, making the integration essential for accurate prediction and personalized care. But the PRS in antibiotic resistance, which can stratify patients based on risk and inform proactive interventions. However, its modest accuracy and lack of epigenetic context limits standard use. Integrating it with clinical and molecular data enhances predictive value and predict who is at higher risk for MDD, major depressive disorder or generalized anxiety disorder, bipolar disorder or schizophrenia. For example, a patient with high PRS score for depression may benefit from early lifestyle or psychosocial interventions. But the PRS can indicate likelihood of response or resistance to certain medications, which include SSRIs. Of course, combining the pharmacogenomics data as well as the PRS data and the epigenetic data will help us to select the more appropriate therapy. The last domain is identification of utilization of CRISPR or Cas9 technology in precision psychiatry. And CRISPR stands for clustered, regularly interspaced short palindromic repeats. It's a natural defense system used by bacteria to protect themselves from invading viruses like bacteriophages. It works by remembering viral DNA sequences and then cutting them when the virus attacks again. This system actually was adapted by a scientist as a powerful gene editing tool. The CRISPR system uses a guide RNA to direct the Cas enzyme to a specific sequence in the DNA or RNA where it can then cut, activate, and disable genes. The applications of CRISPR in psychiatry is like single nucleotide polymorphisms in pharmacogenes can be identified. It also promote methylation status. It enables genetic and epigenetic profiling. And these are the development of various technologies available in the pharmacogenomic world. And of course, PCR, which all of us learned, and next one is microarray technology, and now Cas13a is used in the identification and as a molecular tool. What is the application of personalized antidepressant therapy? And Cas13a can be programmed. It enables the real-time detection of genes like CYP2D6, CYP2C19, SLC6A4 protein. This supports precise adjustments in dosing and drug selection. For instance, detecting low SLC6A4 expression could suggest poor SSRI response warranting alternative therapy. In fact, our team is preparing the diagnostic tool for the SLC6A4 gene protein pattern patients. And this is the model which we have developed it for the identification. By giving one simple drop of blood, we can identify whether the patient has SLC6A4 protein or not. And it's a diagnostic tool, we are developing it. The detecting of low SLC6A4 expression couldn't suggest poor SSRI response warranting alternative therapy, but paper-based CRISPR strips, that's why we are developing it at the department. Using Cas13a and reporter molecules can visually indicate gene expression levels. These affordable portable tools are valuable for monitoring treatment response for screening of low resource settings, especially in India. Otherwise, what we should do, we have to take the samples, send it to the genomic lab, they do the testing, and then they'll give us the test. Like how we are checking the blood sugar level, like self-glucose monitoring. Same way, we want to develop the tool for a mood disorder patient or schizophrenic patient or TRD patients. We can take one drop of blood and we can check whether the patient has SLC6A4 or not, and through that we can initiate the therapy. And the toolkit, what we are developing at the department is has 4 different components. One is phylogenetic risk score, which I explained. Through the GWAS we can get the data, and we have to develop the population pharmacokinetic modeling, and through that we can fix up the dose of the individual drug. It may be an antidepressant drug or SSRIs, non-SSRIs. And CRISPR-Cas9a developer will give you the SLC6A4 data and pharmacogenomics data. So based on this 4 integrated precision information, we are developing the toolkit for the identification. And of course, once it is developed, definitely we will be patenting this technology from Jesus College Pharmacy, Ooty. And what are the advantages of this multi-omic approach? Of course, it gives the most current pharmacogenomic tools, which are static, relying solely on DNA level data. This toolkit, what we are going to develop, integrates both static and dynamic data. Static data is genomic data, and dynamic is transcriptomic and epigenetic data. And CRISPR-Cas13a provides a real-time gene activity insights, which is a game changer for monitoring pharmacodynamic shifts. It also adds individualized pharmacokinetic modeling using population data, enhancing dose precision. So the potential clinical applications of this data is to understand the treatment-resistant depression and generalized anxiety disorders, bipolar disorders for the patient who are not responding for drug response. And if the patient is having first episode depression for proactive therapy, and we can initiate the monitoring therapy, adherence and relapse risk. So this multi-omic approach will help us to identify the TRD patient at the early stage itself, and whereby without doing the trial and error method, definitely we can initiate the therapy at the initial stage, and we will initiate the treatment. And of course, the hospital where I practice is very, very limited resources available. By developing this technology, definitely we'll be able to support the needy patient of our region. So with this, thank you so much for the opportunity. If you have any questions, I'm happy to answer. Thank you so much. Mic, please. To sir. Yeah, in fact, I must thank Dr. Ravichandran who encouraged me to enter into the pharmacogenomics area 50 years before. In a very casual discussion, he encouraged me that enough is enough in pharmacy practice, your own person, but unless we move to individualized therapy, you're not going to be recognized. So thank you so much, sir, for all your guidance and support.
Yeah, thank you, Professor Ponnusankar. It was a wonderful talk. See, the audience, you should understand that this all looks at something new. The new thing always creates challenge, but it gives knowledge. That's the serious concern.
Thank you,
It's
sir.
a very impressive talk. I have a question.
Yes, sir.
For that, the sake of that, our young people,
Sir, please come this side. So that, sir,
How-
please come this side.
how that at the, your the talk which enrich their mind by the way of that few assay system, few test. These are the test. The crisp of that the component is that depressant, antidepressant. So these are the assays need to be learned. These assays would be useful for that assessing the patient. This is what I wish to see as a student. If I learn this at the crisp of this three assay, I can understood about that antidepressant, the cases with that the drug, and this can be that the specialized, personalized component like CRISPR-Cas, Cas13a. Assay component, this is CRISPR-Cas antibodies are available. Cas13a antibodies are available for testing. So how this at the segment of cytochrome, SIB, different cytochrome assays would be useful for this the purpose of that either the dopaminic component or that the serotonin component, the segment of neuronal transmitters with the dopamine, how the very specific, the vagus nerve functional with that the dopamine which regulate. These are the things, few assay system could you say?
Yes, sir. So before that, you please, can you, sir, please say something about how the microbiome and the vagus is very important for the students, because I'm not expert in microbiome.
It's a
So you
very
please-
relevant component. He hinted that basically the microbiome which is present in our nose, which is present in that the gut, gastrointestinal tract and the gut till then that the colon, which has several functions like Corynebacterium, Streptococcus, Lactobacillus, Bacteroids, Faecalibacterium, Veillonella. The microbiome, what it is listed, it has each component. Bifidobacterium, Corynebacterium which is present in the nose, which controls all the pathogen enter from that atmosphere to that inside the body. Whenever the Corynebacterium load is that comes down, the individual get allergy, rhinitis. This is the problem of that the pain. The reason on that, what the food you consumed doesn't balance that the microbiome, so that create that imbalance of the microbiome that causes that allergy. That's the reason that the load of microbiome in the nose, Corynebacterium, if it is differ, the allergy comes, immunology goes down, your liver function goes down. Till then that the load of the microbiome balanced in the gut and reflects in the nose, then that the allergy become normal or recover from that the problem. Basically what I am trying to say that the food what you are consuming, it is metabolized by the help of that the microbiome only. Though the liver enzymes are that supported for this, bile acid, bile corresponding derivatives are that influencing for the metabolism. The microbiome, villanella, which convert the complex carbohydrate material into it's a simple material. Carbohydrate converted into simple glucose and pyruvate. Fatty acid converted into what that the fat you consume as the ghee, oil or any other component of that the fatty material which is converted into carnitine. Carnitine, it goes into that citric acid cycle. Then it goes to that energy level by mitochondria. Mitochondria is that important factor which convert that fatty acid into that energy level. That is a carnitine. Otherwise it stores as a acyl carnitine. These are the very important factors aligned, associated in the metabolism. If the metabolism failed with the microbiome failure or microbiome imbalance, that is what it is a metabolic disorder, insulin resistance comes, metabolic disorder of diabetes and the fatty liver, it comes into that the picture. This is the problem. The problem, how to restore that or address the problem, you have to select the question. Yes, I want to understand that, how that the liver function, it ensure that the metabolism. In the liver, there are 8 lobes are there. Understanding the 8 lobes, you need minimum 8 years. It is not that simple to understand that the liver and liver function, anatomy, physiology and the pathology. If you understand that only the liver and liver function, you will become a big scientist. I can say that if you understand one cell and cell content, cell function, cell imaging, then you will become a big scientist. One simple cell, it is that what it is. Simple cell, it is one segment only, what it is there. A person who is that the top scientist, who knows that only that about mitochondria. He don't know anything. If you ask anything, no, I don't know anything. Only I know about the mitochondria, mitochondrial dynamics, how the mitochondria works on the metabolism, how it is the mitochondria works on that fatty acid, how that mitochondria it works on that conversion of that fatty acid, how that mitophage it is there, how that mitophage and autophage it converts on that, how that mitophage which is connected with that metabolic disorder. So he doesn't understand more than that the mitochondria, he knows only the mitochondria. What you have to understand, what we should know. I want to know what is autophage. What is that the macrophage? Correct? So these are the very serious component. Now I started because we start one component of a microbiome, it goes that keep on continuous with that the several question it would be connected. So I request Professor Ponusankar for that the simple component, 1 or 2, these are 2 assay as a undergraduate. You learn that this is a 2 assay. It gives that another 2 assay lead on that. Thank you.
Thank you, sir. CRISPR-Cas9 is assay which we do regularly to understand the protein sequencing. Of course, it is a little expensive business. There are many private laboratories at Coimbatore, at Bangalore, at Chennai, where if the institution is not having that facility, you can send a sample to them and then they'll be able to help you in identifying the sequencing. So being the research institute, we developed all the facility, and I'm not a scientist, I'm a pharmacy practitioner. I work with the patient every day, and what I observe with the patient is like many times therapeutic failure, whereby many patients, the patient who I met last week, this week he committed suicide. So that is the problem we are facing it at the hospital. And psychiatry department is the one which is very interesting to work, and we are saving, of course, in every department we save the life of the individual patient. The place where I got my training at Australia, where we have elderly population more, and I was predominantly trained in psychiatry area, so elderly population. That interested me to develop this technology here. It's a toolkit, and otherwise when we send the sample, it will be very expensive for Indian population. So when we develop the toolkit, that's what the government of India is also encouraging the people to develop own technology in the country rather than developing on American technology. So I think from now another 2 years or so, we'll be having a toolkit. Maybe it may be utilized for your patient population also at this part of the country. And the assay component is it's like it is developing the technology is not so easy. We have so many practical difficulties, but I'm sure that with the support of many of my colleagues at the institute, where we'll be able to have it. And the research component here is like 4 different components. One is population pharmacokinetic area is one research area where if anyone wants to learn more about that, they can do some research in that area itself. And genomics is another area, and then PRS is another area, epigenetic changes is another area. So, sir, what now explained about the microbiome research. So we have, beyond the pharmacy practice research, there are so many research areas available where if you learn that concepts, definitely you have a very bright future. Of course, we didn't get that opportunity. At this age, we are learning it. And we are teaching to our students. So yes, sir,
Rightly, Professor Ponusangar, what he conveyed, that after 30 years exposure with that postdoctoral fellowship and PhD fellowship with the different country, he is saying that I am learning. Still, I am also learning on that. I tell you one component that what currently, where that with the target of 100 days, I was thinking that I should get 100 crores. If I am not getting 100 crores, I am not worth on that. I got it. 100 crores with 100 days. Our target is that it is not that simply coming and sitting there in Delhi, what we are going to do. Everyone scared about that, what we are going to approve and get into that. This is what it is, the challenge, what we gain. This basically, I am the part of that, the component. With that, the microbiome, what it is there, it is a life. What the current practice it is going, it is only that symptom-based therapy, symptom management. But what is the cause for the symptom, it is not addressed currently in that any of the health science area. That is what it is, a liver and liver associated problems. This is the liver associated problem. Liver dysfunction causes the disease. That is a symptom now currently treated by using that few pharmaceutical drugs. But what is the cause for the symptom? That is what the question we have presented for the project, and we got that 100 crores in the first round of 36 universities participated. We got that our university and JNU, Punjab University, Cotton University, together we have distributed the role and responsibility based on that expertise and facility what they have that, and we have presented our project and we got 100 crores. Within that 100 days, what we committed and what we conveyed, we got that, and we have started working on that. Basically, how that the symptom can be addressed and cause for the symptom could be addressed by addressing the liver function. Liver function damage it comes, how it damage comes, it's a variation in the microbiome, load of the microbiome changes, liver function, dysfunction it happens. Liver dysfunction it happens because of the microbiome variant, or that load of the microbiome comes down, liver function comes down. Metabolic disorder starts. That is a cause for the various problems. These the problems need to be addressed. Mechanism should be that it is suggested to the clinician for the practice, for the better practice and therapy in the health sector. This is what it is a component. So unless otherwise, if you take the question and address the question, you will not get that happiness. All it is comes under that, the end what it is? Laughing. What is laughing? That is a happiness. Yes, I learned this thing today, I am happy on this. Every day if you learn that one segment, one line, 10 days if you learn the 10 segment, if you know that the 10 segment to talk 10 minutes, that is a happiness. You can talk about that, the RNA for the 10 minutes, that's a big happiness. Is it or not? That is a transcription. If you know that Pharmacogenomics, you talk about 10 minutes pharmacogenomics. That is a big happiness. I know the credential of that pharmacogenomics. I can talk about pharmacogenomics 10 minutes. That is a learning you all should have. That gives that wonderful confidence for you. Thank you. Thank you, Professor Ponusar.
Thank you, sir.
Thank you, sir. We will take a short break. Please use this time to stretch, refresh, and enjoy some light refreshment. We will reconvene in 15 minutes and continue our program. Repeat, we will reconvene in 15 minutes and continue our program. Thank you.